All right. Good morning, everyone. So today the goal for this class is for us to finish going through the lecture of the split plot. So we create the full intuition of what it means, what the restrictions are for randomization, how we're assigning treatments to different sizes of experimental unit. Look into the ANOVA because it's going to be a difference there that's really important for us to make sure that we follow. So our model represents what happened in the field. And then we're going to go through a randomization exercise. So I want to show you how you would randomize a split plot in a randomized complete block design. And then we're going to do an analysis. And I hope that we're going to have enough time to go through all this today. And then we can move on to our next topic, which is going to be repeated measures. I think that's probably something that a lot of you have to some extent in your research. And so we want to cover that as well. And then after that, we're going to start getting in the transition between the experimental design side of things and more on the machine learning side of things. So we're going to start off with some regression techniques, especially the regression models to find optimum levels. And then we're going to be downloading some open source data, which is part of the data science side of this course, and using that to create new variables and then start training models to start creating some of that intuition of what is the main difference between experimental design models and machine learning models, and some of the concepts around machine learning that differ from experimental design. Okay, I think we stopped somewhere here. I do want to take a few steps back and quickly zoom through these. So we have already talked about most of this lecture. I just want to touch base in some of the concepts here. So we're still talking about the same study as far as what we're interested in. So our question still remains, the effect of different nitrogen and potassium fertilizer rates on yield, let's say. That's what we've been analyzing. So we have these two factors, N and K fertilizer rates, different levels within them, for a total of 9 treatment combinations. So we talked about when is a split plot needed. So a very typical, it doesn't have to be only this, but a very typical case is when the levels of one of the treatment factors that you're applying require more area. That's normally the case than the levels of the other one. So we went through this example here, assuming that the levels of the potassium treatments are applied with this equipment here, which applies.
Your screen is paused, screen sharing.
Oh, how did that happen? Let's see. Sorry, are you online? Can other people see now? I think you were, I think my screen was frozen for you. Can you see, like, if I move my slides now, can you see that?
Yes.
Okay, awesome. Sorry about that. Well, I guess we didn't talk much about, a lot of things yet, so it didn't miss. But I guess the main point here is that we're looking at this slide and just with this concept that normally we use split plot when one of your treatment factors need a larger experimental area or more experimental material than the other factor. In this example here, we are picturing that with applying the potassium fertilizer rate. So potassium normally is a dry fertilizer that we apply with a spreader, which we see here in this picture. And for that spreader, the swath of application that it has, so the area that it covers when it's applying potassium in this case, is going to be of 12 rows. So we need 12 rows for this treatment here to be applied. Whereas with nitrogen, we're still applying the same way that we were before with this 4-row equipment here. So one of our treatment levels requires 12 rows, the other requires 4 rows. How do we accommodate that? One way that we could accommodate this is simply by having all of our plots being of 12 rows and then just having the potassium rate applied, and then we come back and apply the nitrogen rate in 3 passes per plot, right? So that could be an option that would still have us in an RCBD without a split plot. However, we would need, so the blue rectangles here are comprising 3 4-row passes, or in other words, 12 rows, right? So if we require this much area here to apply one treatment or one rep of the levels of potassium, then we end up needing this much area to cover 9 plots. But let's say if we don't have this much area, right? So if you go to your farm management or your farm staff and ask for this much area, and they say, well, we don't have that because you asked us originally just for the one that we're seeing behind the or in front of the soil types. So if you need more area or more material, but you don't have that material, and that material is only required for one of your treatment factors and not for both, that's one of the cases where we would use split plots. Another very common example is if your treatment structure has irrigation levels. Normally, irrigation levels are going to be applied to a larger area than anything else, right? So maybe you're going to have irrigation by nitrogen, where irrigation is going to be the larger plot, and you're going to have multiple nitrogen levels within them. Or perhaps variety, or perhaps other things that don't require a lot of area, but irrigation does. So normally that's also a very common example in agriculture. So imagining here that if we did have all this area here, so imagine all the blocks being the same length of block 1, and if we did have all that area, we could still fall back into our RCBD. That would be fine. We did not have to have 2 different sizes of experimental units. But assuming that's not the case, assuming that we have limitations in space and we need to be confined within the area of our original study, which is in front of the soil types. So that case is when a split plot would be very helpful for us. So if we're still having our 2a factorial, the way that we refer to every time you see or you read about a split plot, you should expect the person to explain what is the whole plot treatment factor and levels. In our case, the whole plot treatment factor is potassium because potassium is the one that requires a larger area, right? In our case, if I told you all this, that potassium requires 12 rows, nitrogen requires 4, and I tell you the whole plot treatment is nitrogen, something's wrong, something's not matching. Your whole plot treatment factor should be the one that requires the most area or resources. And in our case, our split plot treatment factor is our nitrogen fertilizer rates. We still have all the same 9 combinations. We can still estimate main effects and interaction just like before. There's going to be a key difference in how the error terms are defined. So I guess, so you could have a split plot in a CRD. It could be the case. I want to show you how to randomize and analyze a split plot in an RCBD. First, because it's the most complex of the two. So if you know the most complex, you're going to have an easier time downscaling the complexity to a CRD if you have to. And also because of probably, if you're doing field research, you're most likely working with RCBD rather than CRD. So there is the main difference here that begins on the planning side is how we randomize these. So the first thing we do is we're going to randomize the whole plot treatment levels within each one of the blocks. So it will look like something like this, where each one of the colors here represent a different potassium rate. So we come to block 1, we randomize this much area to each one of the 3 levels of potassium. Notice again, potassium requires 3 of these cells. And that's why we randomized the whole combination of chunks of 3 cells, right? So we come and randomize first for the block 1, and then the block 2, and 3 and 4 independently, just like we did for the RCBD, the previous example that we were doing. So that's the first randomization, is you first randomize the whole plot treatment levels to your plots. In this case, plots of 3 cells, assuming 4 rows per cell, 12 total rows of crops in the field per plot. After you do that, you come back to your study, and now you randomize the 3 levels of your split plot treatment factor, which in this case are nitrogen, depicted by the different green shades here. You randomize them within each one of the whole plot units, right? So you come here on this first unit, so the first potassium rate in block 1, randomize here, another randomization here, another here, and so on, right? So you have many more randomizations on the split plot level than you do at the whole plot level, but it should be that way for you to configure a split plot well designed and planned. I want to show you the model here. I know that sometimes looking at these equations and trying to understand what they mean, it's not the most appealing way of understanding what's going on. but I do want to point out some things here that you're gonna see on the ANOVA, and that's the main reason why I'm showing you that, because your model, what we specify here is gonna be what shows in your ANOVA table, and it's gonna be important to understand. So your YIJK is just the observation, let's say the yield observation that you collected in a given plot that received a given nitrogen rate, potassium rate within a given block. So this is still very similar to what we did before. Mu is our overall mean. We have this, I don't remember, I don't know how to say this in Greek alphabet, but let's just call it P. Does anyone know? Rho, thank you. So rho K here is the random effect of the kth block. So here's a block random effect. So we're treating block as random in this case. And this is a really interesting point because now, because of the complexity of the air structure, it's going to be better to treat your block and interactions with block as random, just so the air is properly assigned to the correct experimental unit. So in this case, your block is being treated as random. Then we have alpha I being the differential effect of the potassium rate. And now notice that, so alpha and rho, so potassium rate and block, the interaction between them is actually being treated as the error of the whole plot level. So every time that we test something related to potassium, is going to be, the error is going to be coming from this interaction here. So this is our first error source of this model. Then after that, we have our beta J's, which are the differential effect of the nitrogen rate, which is a fixed effect because it is our treatment. We're interested in that mean. We have that interaction between potassium and nitrogen, which is depicted by the alpha beta IJ interaction. And then everything else goes to the residual error. And this residual error is then what we use to test the nitrogen effect and also the interaction effect. We're going to see this better when we get to the ANOVA, but just to picture this in your head, that because we have two different sizes of experimental unit and we are randomizing them in separate steps, that creates variability that's attributed specifically to either the larger size or the smaller size experimental unit. Because of that, we have two levels of error. The split plot here is the most common approach that you would see, but you could progressively add more splits. So I had in my PhD a study that was a split plot design. So I had like 5 vectors being split from a very large plot to begin with and all the way to a very small one, all in the same study, meaning that each split that you add, you have a new error. And this is really important that we specify the correct models to represent that, right? Okay. And then our residual, I guess I forgot to bring that up here. So let's look again at this ANOVA, just to remind you. So n, lowercase n is the number of levels in nitrogen rate, which is 3. Lowercase k is the levels of potassium, which is 3. R is the number of blocks, which is 4. And I want to call your attention to one thing here. Up until now, I've been calling blocks and reps interchangeably. Now that's going to change because we're going to have different number of replicates depending if we're talking about the whole plot or the split plot. So I want to just make that distinction here and actually tell we have 4 blocks because that is regardless of the treatments and how they're assigned. But just so you keep in mind that now we have two different number of reps depending if we're talking about the whole plot or the split plot. So that's why I'm changing the syntax a little. And then uppercase N is the total number of observations, which remains the same, 36. Okay, let's walk through slowly on this ANOVA table here and see what is going on. So we start out with our block effect, which is, it has 3 degrees of freedom, just like before. We have our potassium rate, just like before, we have 2 degrees of freedom. And then we have this interaction between potassium rate and block, which is right here, and that is our whole plot error. So if you something to notice, number 1 is our whole plot error has 6 degrees of freedom only. And if you look into the So if you recall the mean square column, the MS column here, the mean square is just the sum of the squares of that line divided by the degrees of freedom of that line. So we have the mean squares of potassium, the mean squares of the air. And then if you look on the F ratio, we're testing the potassium rate effect, so the mean square of the potassium, with the mean square of the whole plot error, which is this one coming from that interaction between potassium and block. So to test the effect of potassium rate, we're using an error term that has 6 degrees of freedom in this example. That's going to be one of the things going to be different and now I guess for now I'm going to pin that, and when we get to the end of this ANOVA, I want to unpin and bring this back up again. You can kind of think of this as like its own study almost. Like if we were just looking at potassium rates, this would have been your study ANOVA, right? But now we're coming inside of those whole plot units that we have for potassium, and we're bringing nitrogen there. So we have nitrogen rates here, so the same degrees of freedom, sum of squares. We have the interaction between nitrogen and potassium, the same as we did before. And then we have the split plot error, which is in this case, is just our residual. So something to notice here is the split plot error degrees of freedom is 18 in our case. You would always expect to have more degrees in a split plot. You should always have more degrees of freedom in your split plot error term than in your whole plot error term. Always, right? Because if we just quickly go back to that, to here, you can kind of imagine that you're only replicating your whole plots. I mean, in this case, each block only has a given whole plot treatment level one time, right? So block one here, let's say that this is the blue here, let's say it's potassium 0, 30, and 60. It may not be that, but just imagining that. Whereas your split plot, you have a lot more. So within this block one here, each one of your split plots are appearing 3 times. So because you have a smaller area, therefore you have more replications of that split plot when compared to the whole plot, that also should be reflected on the ANOVA and especially on the proper error level, the proper error degrees of freedom. So whole plot error has 6 degrees of freedom, split plot error has 18 in this case. Also, something else to notice is we can still estimate potassium rate, nitrogen rate, and interaction effects. It's just that in this case, potassium rate is going to be estimated at a lower power compared to the nitrogen rate or the interaction. Because again, we have fewer observations and therefore fewer error degrees of freedom on the whole plot level than at the split plot level. With all that, another thing for you to notice is when we're testing the whole plot treatment factor, which in this case is potassium rate, the F ratio is based on the mean squares of the whole plot error, which again is this one here. When we get to the split plot treatment factor, which is nitrogen rate and then the interaction between nitrogen and potassium, these two terms should be tested by the mean square of the split plot error, which is this one here. So if you look this F ratio here, in the denominator of N rate and interaction, we have the split plot error. And this, again, is something important to notice because even let's say you plan your study, you randomize properly as a split plot or CBD, and then you come to collect your data, and then you come to analyze this data and you do not specify the proper model, it means that, so let's say if I erroneously run a model exactly like we did last week for an RCBD, what happens is that your degrees of freedom of the error are the same for all of these effects, meaning that you're probably going to be over or making more powerful the effects of potassium rate than it should be, and making less powerful the effect of nitrogen rate and the interaction they should be. And again, I'm saying they should be based on what you plan or what you actually implemented on the field, which is what should guide your decision of what model to run, right? The model that you run is the model that you plan for, the model that you randomize based on the actual field. That's the one that we should be running. So I have 2 notes, I guess, just to reemphasize. We have 2 error sources, the whole plot error source and the split plot error source, and they are used differently here to test different effects. And I guess we just talked about here, like what would happen if you come here and you actually run this as an RCBD without a split plot structure is you're going to be, so your errors, so it's basically going to combine these two errors into one, and it's going to test everything with that same error, which should not be the case because you randomize in a way that you had two different sizes, you had two different experimental unit errors that we should account for.
I have a question.
Yep.
You can also do it otherwise, like, you can do whole plot with N. Or it's the standard one.
No, so really the whole plot should be whichever treatment factor requires the most area, right? So in our example here, potassium is at the whole plot because we are assuming that it requires 12-row plots and nitrogen requires 4-row plots. So that's why potassium is on top. If for some reason it was different, so let's say perhaps we go to our farm, research farms, and then you have a nitrogen applicator that's actually 12-row wide, and then you have a potassium, not as, so there's a different way of applying potassium that instead of spreading the dry fertilizer, it actually just applies right on top of the rows, like right by the row. That is an equipment that exists. So if that was the case, if your nitrogen actually require more area than the potassium, nitrogen should be at the whole plot level, and then potassium should be at the split plot level.
But it will not also depend like what's you are more interested, because in this you are giving nitrogen more power.
That's a good point. So Aminder is asking if, so in this case here, we're given this, I mean, every time that you're on a split plot, your split plot treatment factor is you're gonna have more power to find differences at your split plot level compared to your whole plot level because of that difference in replications at the different levels and also the error that is associated with each one of them. So if you, let's say if our main interest was in potassium rate, I mean, the problem is then, so let's say if we just go back here and we think about that, if our main interest was in potassium rate, but still potassium rate had to be applied on this size of experimental material, you either would need more area to run like a study that maybe you had more replications with potassium. Or you would have to find a way to apply to a smaller area that you could still be within, the limits that you have. So it's more, so it is an important aspect, but at the same time, you cannot just flip because one is more important than the other, right? The decision here of which one goes on whole plot is a lot of times, most often based on limitation of experimental material, right? So even though if you say, okay, the most interesting factor for me in this study is potassium rate. I wish I could have potassium rate at the split plot level. Okay, that's a wish. But if you don't have the experimental material to implement that, then you're stuck there, right? But of course, if you have I guess if you have the option of choosing which one goes where, then you really don't need a split plot, right? Then you can just go with your regular RCBD that we ran. Because then you don't have the limitation of experimental material. And really, we're going to talk about this, I guess, towards the end of this lecture here, but a split plot is to account for limitation on experimental material. If you can avoid it, meaning that if you have enough experimental material to run without this structure of whole plot, split plot, you're better off doing that. Because even though, so there are, I guess I'm going to say this in words, but I'm not going to show the proof of this, but there is a proof in stats material I can share with you if you want to see that. There's proof that the overall efficiency of a split plot is efficiency, meaning the power of a split plot to find treatment effects and differences overall is going to be reduced if you compare with an RCBD that does not have the split plot structure. In specific, and that overall reduction in power happens because you're compromising a lot of power on your whole plot treatment factor. So your whole plot treatment factor, you're going to be estimating everything using this model with less power than if you would having everything in the same hierarchy level. So no split and whole plot. Even though your split plot is going to be estimated with higher precision or higher power, your split plot factor is going to be estimated at higher power than if you were just an RCBD. So you gain some power in a split plot compared to just an RCBD without split plots and RCBD with split plots. The split plot factor and factor, you're going to have a little bit more power than comparing to a non-split plot study, but you lose a lot of power in the whole plot compared to not having split plots study. In a way that when you compare the overall power of a split plot design is going to be less than a design that does not have a split plot structure. So if you can avoid it, because you do have enough experimental material, it's better to avoid it. But normally when people use split plots is because they cannot avoid it. They have these space limitations that one treatment factor needs more area and the other needs less. Just skip over here. So just to reiterate this point again, if you knew that you needed a split plot, you designed a study as split plot, you implemented a study as split plot, you collect your data, and then you come in R and you specify the wrong model. Let's say you specify an RCBD model, you are going to be running the wrong model, right? So all of the assumptions based on how you randomize that study and how that has implications on independence of observational units, of experimental units that the ANOVA requires. If you run the wrong model, you're basically overseeing all that and making your model, again, less reproducible, right? Because you're not running the correct model for what you actually implement in the field. So it's important to know how to do that. So on the, this is just an output from R. So you can see that really on this ANOVA, there is not, there's really no difference from the ANOVA when we look at the R output from when we just ran as an RCBD. Because again, the degrees of freedom that it shows are the numerator degrees of freedom of the MS ratio. So are these ones are the numerator of the, so I don't know if we talked about this yet. So let me just go a little bit slower than I was anticipating. Every time you look at an ANOVA, if it just shows you one column for degrees of freedom, that is going to be the numerator degrees of freedom column. What I mean by numerator is going to be the degrees of freedom associated with whatever goes on the numerator of the F ratio, which in this case here, MSK. MSK was calculated using degrees of freedom of potassium, which are 2, right? So this column here are showing the degrees of freedom of the numerator. Sometimes some functions in R may give you 2 columns of degrees of freedom. One's going to be the numerator, the other one's going to be the denominator. For some reason in my R, I could not make it work to show the denominator. I tried different packages, functions, I cannot get it to work. But I remember that I have done this in the past when I was working with that crazy split plot study, that I could see the denominator degrees of freedom, which if it does show you that, you would expect on a split plot, your denominator degrees of freedom should be different if you're talking about the whole plot or the split plot level. If you're just running RCBD, they should all be the same because you only have one error source that's testing everything on an RCBD without a split plot. With a split plot, you should have different denominator degrees of freedom that are telling you different error terms are being used. Yeah, I think I talked a little bit about that here. And then this trade-off here, I guess I jumped ahead of myself, but if you have an RCBD that is not randomized as a split plot, like we have done in the previous example or the previous exercise, comparing that RCBD, so I'm just calling that RCBD, with an RCBD of split plot, the whole plot treatment factor has power decreased. When we talk about power in statistical terms, we're just meaning being able to find statistical differences, right? So your ability to find statistical differences in the whole plot is decreased when you compare it to a randomized complete block design without split plots. On the split plot treatment factor, your power increases, but overall, combining both of those effects, if you compare split plot RCBD with just an RCBD, the split plot RCBD has less power than its RCBD counterparts. So just a summary here. We use split plot designs when we require different amounts of experimental material. This is most commonly area to apply the different treatment factors in a study, where the treatment factors. So the levels of the treatment factor that requires more area are applied to the whole plot treatment or the whole plot experimental units. And the levels of the treatment factor that requires less area are randomized on the experimental units that are smaller, so the split plot experimental units. And because we're doing this randomization in two different levels, that creates two different error terms that we have to make sure we specify in the model. So the model uses the correct error to test different effects. And again, normally we use split plots because we have to, because if you did not have to, you could just not do a split plot, you would be better off. But if you're limited by area or resources, then we use split plots in that case. And even though your split plot treatment factor power, so in our case, nitrogen has the power increased, but we lose a lot of power on the whole plot treatment factor, which overall makes a split plot be less powerful of a design comparing to just running everything at the same level as an RCBD without having that hierarchy. Okay, so this is the lecture that I had. Before we go into the randomization exercise, I just want to take a moment here and open up for questions.
So, let's consider, a person has, 10 rows. And they apply, on half rows, they apply K, and half rows, they apply N. So in that case, we have to consider whole plot or split plot error.
So if you have 10 rows and you apply one level of potassium on one side and one level of nitrogen on the other side, I guess that is not a proper design, right? So if you, let's just go back to that, to this again. So in your case here, you're saying like, if you come here and then you apply a potassium rate here and then a nitrogen rate here, so you're not creating a balanced structure in that case, right? So here, what we have to do in this case is we're applying one of the treatment factors to a larger area, and that would be your whole plot experimental unit, right? So this would be the whole plot experimental unit. And then you come in here and you randomize your split plot treatment factor. So it'd be nitrogen. So if you think about it, let's just, if we go to this here. So this whole plot here received one potassium rate. So let's say that this was 30 kilograms of potassium per hectare. But now, so this whole blue block received one potassium rate, but then we randomize the nitrogen rates in here. So, you know, this would be, let's say, zero, I don't remember our levels, but let's say that there were 0, 100, and 200, right? And now you do the same thing on the next one, but you randomize again, right? In a way that if you look at this plot only, this plot here had, let's say, a potassium rate of 6. So all three plots had the same potassium rate. Let's say that was 60. So 60, 60, 60, but then the nitrogen rate is changing at the smaller level, right? So in a way that all plots still received a given potassium rate and a given nitrogen rate, but we apply them at different sizes or using different equipment that require different sizes, and we randomize accordingly as well. Another thing I don't want to make this more complicated in your head, but I think it sets the stage a little bit for when we talk about repeated measures. Some people analyze repeated measures as a split plot, where they would have the split plot factor being time. So imagine, you know, I guess if we just forget of what we talked about in this case here, and we just create a new imaginary design here where we have potassium, so the whole plot would still be potassium, but then we had time. So instead of nitrogen, we had time. Let's say we measured plant height at 3 stages during the growing season. So we have, I don't know, maybe 10, 50, and 100 days after planting. Those would be our time levels. Some people do that, and there is a problem with that. Can anyone guess what is that problem? So imagine that now the green cells here are time. Yes, we cannot randomize time, right? I cannot come here and say that, you know, this, because then you would be measuring, because even though here you may be, you still, you know, we may still be thinking, well, I'm going to measure the 50 here, the I don't know what times I said, but let's say 50, 100, and 150 days after planting, you can think that you would still sample from different parts of that field, but really the goal normally is that you sample the same part of the field, right? So you go, let's say on all this plot here, we go to the same center rows and measure height from the same plants, because you do want to track those plants. If you were to sample different plants every time, and the variability between plants may be too large for you to really find the effect of time more precisely. So you would normally come and measure the same plants within the plot. But because I cannot randomize time, I cannot say that I'm measuring the 100 days first and then the 50 days, right? I cannot do that. So the lack of randomization of time or space is what dictates a repeated measure design, which then we have to account for this lack of randomization. In a split plot, if we don't have repeated measures in a split plot, so let's go back to our potassium and nitrogen example here. You can randomize the potassium rates, you can randomize the nitrogen rates, and because of that, this is a split plot, right? Because you can randomize them, we don't have to treat this as repeated measures. But just to create a little bit of connection between this topic and the next one. Okay, any other questions or thoughts about split plots?
If you had another split, what happened with the power of the next split?
So let's say if we were to add another split, maybe one way of doing that is you could cut through the plots vertically, right? In a way that now the nitrogen plots are being segmented into two pieces, like the left and right piece. And then you would randomize another treatment to either left or right in that nitrogen plot, right? So then if you imagine that, so, if we just focus on block one, so block one has a given potassium rate one time, right? So this is one potassium rate, this is another one, this is another one. So we have one potassium rate appearing one time on each block. If we talk about the split plot, so let's focus on maybe this darker green here. So we have a given level of the split plot factor appearing 3 times. So 1, 2, 3, right? So the more splits you make, the more times you're making that smaller split to appear per block, which makes the power of that increase, right? So let's say if we were to come here and do a split plot design with whole plot potassium, split plot nitrogen, and a split plot variety, for example, right? So we could split these plots vertically here, where the left side would receive variety A, or variety A and B would be randomly assigned to one of the sides, right? Yeah, so then you would imagine that it would have variety A appearing, let's say 1, 2, 3, 4, 5, 6, 7, 8, 9 times, right? Because it's appearing once in every nitrogen plots. I mean, in one of the sites, not, I just counted kind of in the same column, but it would be randomized one or the other column. So the more splits you add, the more degrees of freedom of that error you're going to have of that lower split. But your whole plot error remains the same. Your split plot F level, your split plot error remain the same. You're just creating more structure here. Yes.
So based on the random and fixed for the block, we mentioned that number of blocks will be more than 5. So but here, the block number is only 4. So on that case, can we assume random effect for the block?
Yeah, so that's a really good question. So here, what happens is, if we do not treat block as random, the way that the different, so if we do not treat as random, then we do not have multiple variance components being estimated, and we just have one in the end. And the problem with that is we have to have multiple variance components being estimated at the different levels that we have for the whole plot and split plots. And the way of doing that is by treating as random, as we're going to see in the example. So here is almost like a trade-off where we're going to have to sacrifice estimating more precisely a variance components in lieu of actually being able to estimate different pieces of that and use them appropriately. So it is a trade-off. And it's really interesting because, we didn't get at all in the topics of how behind the scenes, how the matrices of the design are changing and incorporating these things differently in the analysis if we run as fixed or random. but they are changing. And so if we do not treat as random here, because that's, you know, that's, it's something that I think let me clarify one point, I guess. So we use the random effects, as we talked about, when we're interested in more of the variance of the factor and not on the means. But also we use random effects to define the appropriate error structure of a model. So if we did not treat blocks as blocks and its interactions as random here, we would not be able to identify the proper error structure of this model. So we use for that reason as well. And really, as we start, and let me just take one step here and tell you something. In experimental design language, this is a split plot, a mixed effect split plot. But if you think more broadly, just in like linear model terms, this now becomes a multi-level hierarchical model, because your errors are coming from different sources. It's not just a single source of error. They're coming from different sources within your design. So this becomes a multi-level hierarchical mixed effect model in that sense. And for that to be properly specified, you need that random effect of the block and its interactions. That's a great question, yeah.
So it's like subsetting the random errors through split plots. Like there is a whole error random error block. So splitting is like subsetting anything
Ye-
that way.
Yeah, so it is subsetting, but in a hierarchical way, right? So if we look at the ANOVA, I guess maybe that gives us more of an idea. So the whole plot error is looking at the variability of potassium rate levels within the block, that residual variability, that in this case is explained by that interaction between potassium and block. And the split plot error is going to be everything else that's not explained by that, or the remaining fixed effect components of your model, right? So we do have these two levels that if you had not randomized like we did in like first the whole plot and then the split plots within the whole plot, if that randomization structure was not there, you would have only one error term that would be the same to test all the effects. But because we did have this structure, and because so by having, let me come back to that drawing again, just to, so if you imagine another thing that the different error sources are doing for us here is that, so the whole plot error is accounting for that variability of potassium rate, but it was all randomized within the block. So it is accounting for this relationship that potassium rate in block may have, and that's it. And then the split plot error is accounting for that, but also for the fact that that you have this limitation where nitrogen rates were randomized within each level of potassium. So it's almost like, for nitrogen, it's almost like potassium is the block. If you imagine that, if you think about that. And so now we have this this correlation between a given potassium rates, I'm sorry, a given nitrogen rates nitrogen rates within a given potassium rate are correlated to some extent because they are all being applied on the same potassium rate, right? And then you go to the next one and the next one. So this creates a potential correlation structure that you don't have if you don't have that restriction of whole plot and split plots. And that is properly modeled by having the different error terms as well. And so it's the fact that you're randomizing at different levels that's creating different error terms, and we need to use the proper error terms so we are running the model that represents what happened on the field. I know this, it's a little bit confusing, and I hope that when we get to the exercise that you may be able to visualize or maybe grasp the concept a little bit better. But really the main takeaways here is that we have different error sources and we have to properly identify them in our model to make sure that we're running the model that represents what we did in the field, and therefore, doing the best science we can. Any other questions? All right, so I don't hear anything. Let's get a setup for the exercise. So I did upload a couple of scripts to our GitHub repo. So if you want to do a git pull on that, you can do that. And after you do a git pull, we're going to go through creating a new project. So if you remember, creating a new project is going to start on your GitHub. So that's going to be your personal GitHub page. I want to call that zero. Let me maybe write that down somewhere so you can have a reference. So I want you to, so go to GitHub, to your GitHub, and create a repository called 05 SPP for a split plot. So you create that on GitHub. As you know, I want you to make this, a public repo. I want you to add a README. So the instructions, if you remember, we have those steps in a different lecture. Let me just go find that to have up in the screen. This is it. Okay, so this would be the first step. You making a repo on GitHub. So make sure you name it 05s_SPP. So it's our 5th project. It's for the split plot analysis. You do not need a description. Make it public. Add a README. Add a git ignore for R, so you can select the language in that drop-down menu, and then create that repository. So this has 3 slides that I'm not going to be able to show you all 3 at the same time. But this, again, this lecture here was posted when we went through Git and GitHub. It's the lecture on Tuesday, January 23rd. That's the lecture that I'm showing you. So if you need a refresher on what to do, you can go on your computer to that lecture. I have here the first step on the screen, so you can follow that. But if you want to, there are 3 steps. So the first one is making the repo GitHub. Then we want to clone that. So I'll show you how to get the path, which I guess you're already familiar with this. And the last step is creating the RStudio project, where we want to follow these steps here to make sure that our RStudio project is connected with our GitHub repo, and we don't have to fiddle too much with that because RStudio does the behind the scenes work for us. In a way that in the end, you should create this in the main folder of the course. So you should be seeing, let me pull up mine here to show you. So you should be seeing your 05 SPP alongside the others in your folder. Once you do that, you can copy and paste the 2 scripts that I uploaded on the class GitHub to the code folder of the 05 SPP. And also you can copy and paste the data that we already used for the 04 RCBD, copy from that on your computer and paste in the data folder. So this is how it should look like. Your 05 SPP folder in your computer should look like this. Of course, I have the completed versions here. You only have the partial.
We will use the same thing, not like it. But we will not use much. No. So go here. Yeah, now you can copy and paste this thing.
Okay, I see that here in Athens, people are working on it, so I'm assuming you still need a little bit more time. Does anyone have any issues?
Maybe change some items here. Oh, because I need help with that.
You are taking me to resident.
You can come. I can bring something from the field. That's in
Yeah, do bring.
that's in my hands.
If you don't, then you don't interview this.
If you can fit in bag, then I can do.
No, it's not good. I can.
No, in big bag. None of them are very. You can fit in there.
Whatever interviewers come back,
So
they will
now want to.
you will be on
So
the
we
suspicious member of
Files.
trying to bring a lot of unwanted substances on the yes. They always give some of the countries and bad beliefs that where they coming from there.
Yes, good experience.
How?
Every time you saw, so he doesn't files, but I guess what happened is. It seems like we need changes. They're actually almost-
Yeah,
He had that.
so that they will take it on.
Yeah, he got some spices from Bharat and they got it back.
Yeah, they check every time by poking holes if you are in that problem. Yeah, every packet they bring, they will just poke holes, even if you bring samples on all the stuff. If you are in that shape,
It's a good. This time when it comes from.
Okay. Have to be.
Anyone needs help?
I have a question about data. I got the codes, so the data that we're supposed to bring in is.
Yeah, so the data is the same one that we use for the 04 CBD. So even like you have that on the GitHub 2024 DSA, but you also have on the 04 CBD. So if you just go back to that folder in the code, in the data folder, copy that, paste that into the data folder of the 05 SPP. Okay, any other problems, or was someone, let's begin here in Athens, was someone in Athens not able to do this, where created the GitHub repo, cloned that, created the code data and output subfolders, did a git pull on the 2024 DSA, copied and pasted the codes inside the code folder of the 05 SPP, copied the data from the 04 RCBD into the data of the 05 SPP. Okay, I don't hear anyone here in Athens. Online, was everyone able to follow these? Okay. Right, I'm seeing some heads shaking yes, so I'm gonna assume everyone was able to do it. So let's get started then. So please go ahead and launch your 05 SPPR project. Just as a reminder, you always want to launch their project and not just double click on your scripts, as that may launch your script in a different RStudio window, and that there's no good comes from that. So always use your projects. Okay. So if you launch that, you may not be seeing this script yet, but if you come on the Files tab, on the R blue button to take you to the home directory of the project, you click on code, and then you can launch your, let's begin with the, sorry, I actually had the wrong one pulled. I had the mix partial. That's going to be the second step. The first one I want to do is the randomization partial. So we learn how to randomize a split plot first, and then we're going to get into the analysis. So make sure you pull up the randomization partial script. Okay, so a lot of, I guess, both of this script and the analysis script, I left a lot of the things that we have already done that are the same for the previous design, so we don't just redo everything from scratch. I did remove the pieces that were specific to this design so we can build them together here. So the goals of this script.
Festus, I forgot to ask. I moved the code over to the code folder.
Mhm.
Was there any data we needed to move over?
Yeah, so the data is going to be the same as we used on the 04 RCBD. So if you go in that one, just copy the data from that folder and paste that into the data folder of 05 SPP.
Yes, sir. Thank you.
All right. Okay, so the goal here is for us to learn how to randomize a split plot RCBD in R, and then create the plot layout with that, just like we did previously. So we can go ahead and run your packages here. So we're just loading all packages within tidyverse. And again, on this body here of the script, what I'm telling you is we're going to be randomizing a two-way factorial like before, but in a split plot design, which is different from before. Right? Before we're having all the 9 combinations of 3 potassium rates, 3 nitrogen rates being randomized as one thing, right? Now we have these 2 levels of experimental unit that we're going to have to randomize separately, as we saw on the drawing. So the first thing that we do randomize is the whole plots to the blocks, as we saw on the lecture. And again, I'm just telling you here that I decided to change to call the blocks as blocks and not interchangeably between block and rep, because now we have 2 levels of reps, if you think about it. And we did not have that before, so I want to call block an actual block and not just rep, so we don't get confused. Okay, so if you just come here on this code, whenever we finalize this chunk, I do want to rerun it with the set seed, so we all get the same results. Until we get to that point, is it okay if you don't, because each one of us is gonna be running different number of times, you know, and it doesn't matter until we finish this piece of the chunk. So again, the first thing I want to do here is take our whole plot from factors and levels and randomize that to the blocks. That's what we're going to do in this chunk. So you can give an assignment symbol. If you remember, we used a function called crossing to get all the combinations of 2 or more different things. So let's use that function here, crossing. The first one is our, what we've been calling K rate underscore KGHA. So our potassium rate in kilograms per hectare equals to, I use the C function, I'm gonna give three values. So the 0, 30, and 60, those are our levels of potassium. I want to create all possible combinations of these three treatments with the blocks, right? So I get out of that C function, give it a comma. I want to break code just to give me more space here. And I want to have a column called block, where this is just a sequence from 1 to 4. So we would specify that with the code. If we just run this and we print this result, we have 12 rows and 2 columns. 12 rows because we have 3 rates and 4 blocks. 3 by 4 is 12. Right, so this is our, what we have going on here. Let's add a pipe. I do want to just have all block 1 observations order first, and then 2, 3, and 4. So that means we need to use the arrange function on the block column. So we sort the block column by block number. So we see, you know, in a confined way here. So what you're seeing is right now we have in block 1, we have each one of the 3 levels of potassium, the same for block 2 and 3 and 4 and so on. And just so you create this idea as well. So if you remember the drawing that I have on the lecture where we first were randomizing the whole plot. So remember that we had the larger rectangles around 3 cells of the plot. This is what we're doing right now, right? And that's why we have just 12 observations. Imagine that our study is just potassium. Right now, we're not considering nitrogen at all, right? We're just talking about the 3 levels of potassium only being randomized to the block. That's all we're doing right here. So we can add a pipe here if you want to come after that comment. So this is our randomization. Let me ask you this, how do we do this last time? If you think of when we did the randomization for the randomized complete block design, which it's going to be very similar here. How do we do that? If I want to randomize the 3 levels of potassium within each of the blocks, what do we need to do? Group by block. Okay, that's the immediate next thing. So let's do that. Group by the block column. We're gonna have to use now a mutate function. I want to have inside of this mutate the potassium rate name, just like before. So we're overriding that column. And then we use a specific function here to kind of like shuffle the levels of potassium inside block. Anyone remember what was that function? So the function is called simple. And then we just say simple potassium rate. So if we do this, within each one of the blocks, we are shuffling the potassium rate levels. So if you run that and you print it, now it should not be 0, 30, 60, 0, 30, 60 as before. It should be shuffled within each one of the blocks. And just to remind you, the reason why we set seed is because each time you run this piece of code, this sample function is that random function that is creating a random assignment or shuffling. So each time you run this, you're going to get a different randomization. And you can try that on your side if you wish. And whenever we are satisfied with our code, we want to make sure we run it, but including that set seed, because that's going to make it random, but reproducible for us. So just to remind you on that. Okay, so we just shuffled it. I do want to add a pipe here. I want to come after that comment, creating the whole plot numbers. So I want to use a mutate function to create a column that's going to give me the whole plot numbers. So remember, we're using 101, 102, 103, all the way to 109. That's not going to work here. I mean, it could work, I guess, but it's not going to be as informative. I do want to create a whole plot number that relates to that whole plot. So that whole set of 3 cells in that map that I showed you earlier. So let's call this W plot for whole plot. And so for this, we're gonna use a similar style as before with a slight difference. So open and close quotation marks. We had the block number times 100. So if block is 1 times 100 is 100 plus 1 through 3. This works because our data set is still grouped by block. So if we do 1 by 3, it's going to know to add 1, 2, and 3 for each one of the blocks within, I'm sorry, for each one of the whole plots within the block. So 101, 102, 103, and then it goes 201, 202, 203, and so on. So now I have my whole plot numbers along with the randomization. I do want to do one last thing here, which is to ungroup this data set. So if you add a pipe, we're going to call the function ungroup. So behind the scenes, we'll remove this grouping structure of this data set. So if you print it, you should still have those 12 rows, 3 columns. The sequence of your potassium rates compared to mine may be different because again, each time that you run this code without setting seed is going to give you a different randomization. And that's okay for now. We're not going to worry too much about that at this point. So just to think about it again, we have 12 rows and not 36 as it was the number of rows on the RCBD that we did, on the previous exercise. So right now we have 12. We're going to have 36 at the end of this script, but for now we just have 12 because it's the 3 potassium rates being randomized to 4 blocks. 3 by 4 equals to 12. Okay, so now that we randomize our potassium rates, let's go ahead and randomize our split plot treatment factor and its levels inside each one of our whole plot experimental units, where each row here is a whole plot experimental unit. So I left you the name of the object that I want to call this. Let's begin by giving an assignment symbol and also using the crossing function. So here's going to be a little bit different, and I want to just help us to develop the intuition behind what we're doing. So if you think about it, on the step above, we wanted to randomize potassium rate within each one of the blocks. So we had to have a crossing between potassium rates and blocks. Now, the goal next is going to be to randomize nitrogen rates within each of the whole plot numbers. So imagine that so we're going to have, in the end, we're going to have the plot 101 appearing 3 times, 1 for each one of the nitrogen rates, right? So let me say again to kind of make it more concise. When we randomize the whole plot treatment levels to the block, we need a potassium rate and block. Now I want to randomize the split plot levels, which is nitrogen, to the whole plot numbers. What do I need to have in the crossing? Okay, N rate. So let's begin by that. So we need N rate, KGHA. We use the C function, and the levels that we had were 0, 100, and 200. I want to get outside of the C function, give it a comma and break code. So I want to randomize nitrogen levels inside the whole plot experimental units. What do I do here next? Okay, so we can call this W plot. Where is that coming from? From here, right? I have a column here that's the whole plot number. So I want to copy that object name. So the whole plot, that W plot column is going to be all the levels that I have of the whole plot from the object above. So the whole plot would be the WP RCBD rand, which we created above, $sign W plot. That's the column that has the whole plot information. So if you run this and you just print that, now we should have 36 rows because we have 3 nitrogen rates and we have 12 whole plot experimental units. Let me just show the code in case we're still catching up. Just to, I guess, facilitate on our visual assessment of this, let's give it a pipe and let's arrange by whole plots. Right, so now what we see is whole plot 101, which again, 101 received a 1 potassium rate from above. If we go back here and check, in my case, 101 received 30 kilograms of potassium. So now plot 101 that received 30 kilograms of potassium was further split into 3 plots, right? Where each one of the subplots or split plots are receiving a different level of nitrogen. However, the way that the crossing function does is it just does 0, 100, 200, 0, 100, 200. So that is not randomized. It has not randomized nitrogen levels to the whole plot yet. Okay, so that's what we're going to do next. So let's add a pipe. If you want to come after that comment there, maybe someone else that has not helped me yet. What do we need here to randomize the nitrogen levels to the whole plot? Okay, Pramod is saying group by what? Whole plots, right? So within, and always, I think the word within whole plot is what should trigger you to think group by whole plot, because we want to do something within whole plots. So we group by whole plots. Let's call again this mutate function as we did before. We're going to say nitrogen rate, kg per hectare. What was this? What was the function name again to shuffle things? Sample, right? So sample nitrogen rates. So if we just run at this point, and then you print your object, now those rates are shuffled within each one of the whole plot units, right? So now I have 0, 100, 200, 200, 0, 100. Notice here that I will never see the same nitrogen rate twice in the same whole plot, right? So let me say that again so this sinks in. I should not see here the same nitrogen rate, so let's say 0. I should not see 2 zeros within the same whole plot because I am grouping by whole plots and randomizing all 3 levels. If I were not to group by whole plots and just leave the randomization nitrogen to be unrestricted, I could end up having 2 zeros together in the same whole plot, which is not a split plot then, right? Remember, these concepts are based on how we restrict randomization. And that's what makes this what it is, or partially that's what makes what it is. Also the fact that we have different sizes of experimental units. But when it comes down to the randomization aspect itself, it does not, like R doesn't know that we have 12 rows, 4 rows. That is all context that we bring into it. Our main goal here in the code is to know that we have to randomize the whole plot to the blocks and then the split plot to the whole plot, right? So that's the intuition that we're transferring to code here. Do we need to set seed in this chunk? So Sekar, what we're gonna do is, for now, we're not gonna be worrying about setting seed. Because whenever we get to the end of this exercise, I want everyone to rerun from the first randomization. So if we set seed there and then we run everything once, we should get to the end with the same results. Okay, so the next thing I want to do here is I want to create a split plot number. So it's going to be a new column. So let's give it a pipe. I want to come after the comment here. We're going to use the mutate function to create the column called splot for a split plot. So this name, the way that at least I want to create this is the following. So if I come here on the whole plot 101, I want this specific plot 101 that receives 0 nitrogen in this case to be 101 dash, or yeah, 101-1, 101-2, 101-3, 102-1, 2, 3, and so on. So I just want to add a dash and a 1, 2, or 3 after it to represent what's the split plot number. So for doing that, we're gonna use a function called paste zero, which we use to combine strings. We have used this in the past. The first thing I want in this paste zero is the whole plot column. So we did that. I'm open and closing quotation marks to give it a dash. Add a comma, and now the last piece is gonna be 1 through 3. So again, this works because we group by whole plots, and then we end up having each whole plot appearing 3 times. So that's why doing 1 through 3 here works, because it's still grouped by. So if we run this now and print, this is what we have. So we have the whole plot, which is 101 still for all those 3 cells. But now we have a split plot ID, which is that 1011, 1012, 1013. And then lastly, I just want to ungroup this. So give it a pipe and let's call ungroup. So if you're running now, you should have 36 rows with nitrogen rate column randomized, with the whole plot column and the split plot column referring to the IDs of those plots. All right, so now if you think about it, we have, so here on this object that we just created, we have the whole plot, the split plot, and the nitrogen rate. And then we have the potassium rate on the object above. So I want to combine both of these. So in the end, we have 36 rows, but I know exactly what was the nitrogen rate, what was the potassium rate, what are the plot numbers. So we're gonna have to do a join here. If you go to the next chunk, I left you the name of the object that I want to do the join, or that I want to name and assign the join to. So let's start with the assignment symbol. The first thing we're going to begin with is the first object that was the WP RCBD rand. If I just print that below, that's the one that has only 12 rows, and it has potassium rate, block, and whole plot only. Now I want to combine this with our nitrogen randomization that we did, but I do in the end want to have 36 rows and not just 12. Because of that, we're going to use a slightly different join. So if we give it a pipe, we're going to use a right join. In the past, we've used a left join. So what the left join is it looks on the data set on the left, which in this case is the first one that appears. So this one. And then it looks on the one on the right, which is the one that we put inside the right join, and then join them with the frame being the first one, being the one on the left, right? So if that frame starts with 12, your left join is probably going to give you 12 rows. In this case here, we're using the frame, the one that has 36. That's why we're giving the right join. So the one that has 36 rows was this one that we created above. So that's the SP underscore RCBD underscore rand. If we run this and we print that, we should end up with 36 rows where I do have potassium rate, block, and whole plot that came from the first object, and then the nitrogen rate and the S plot that came from the second object. And we should have 36 rows again because we use a right join. If we use a left join, something else would have happened. And then lastly, on this chunk, I just want to create that treatment name column that we've been creating a lot of times. So you add a pipe, let's call it mutate. Name of the column is TRT name. It was just a paste zero as well. So we do a paste zero of, in this case here, I think in the past, when we did treatment name, we were doing nitrogen rate plus potassium. In our case now, I want to do potassium first because that's our whole plot, right? So let's do potassium rate first. And then we give it, we give it a comma, quotation marks, we add a plus, comma, nitrogen rates. Just so we allow our treatment name to represent that structure that we have in this split plot as well, as far as potassium rate first, and then nitrogen rates. So this is what you should have in the end. 36 rows, 6 columns. These are the columns you should have. Okay, so before we move on to plotting, I just want to pull out block 1 for us to look into it and perhaps understand again what is going on there. So if you had, so this object I left you on this chunk is the one that we just created above. If I just want to look at block 1, let's just do a filter for block equal sign to 1. Okay, so notice now I have 9 rows, right? Because I have 9 split plot experimental units. I have only 3 whole plot experimental units. So if you see a 101 repeats 3 times, 102 3 times, 103 3 times, because that was the level that received potassium, and you also see the potassium rates repeating. So the potassium rates were randomized at the whole plot level, and that's why they get repeated when we repeat the whole plot number. So we should not see here within the same whole plot number, we should not see different potassium rates, right? Because potassium rate was randomized at the whole plot level, so we should only see one potassium rate associated with the whole plot level in this, or a whole plot experimental unit in this case. Now the nitrogen rates were randomly assigned within each one of the whole plots, so we should see them changing. So 200, 100, and 0. I mean, it's kind of interesting that two of them were the same randomization, but the last one was different. In my case, at least that I'm showing you, which again may be different in your case because we have not established the set seed yet. And then finally we have the treatment name. So this is how the block 1 looks like. So block 1 has 3 whole plots, 101, 102, 103. Within each one of those whole plots, we have further 3 split plots, in a way that in total, we have 9 split plot experimental units within block 1, which represents what we saw in the lecture as well. And this again is just a sample, or not a sample, it's just pulling block 1 for us to inspect, but the same happened for all the other blocks. They were randomized independently of this, just to remind you. Okay, any questions here? So if we come on the next, so this is your randomization. This is where things stopped as far as statistical planning of this design. The next chunk is just adding some more columns to be able to make a graph of this, right? So if you come to the next chunk, I left you everything there. So we're starting with the object that we just created above, just creating the rows and the columns so we can show these in ggplot. These rows and columns have nothing to do with the design. These are just for visualization purposes. So I left you all the code there. I think you can just go ahead and run it. Since we have already developed this on the previous, or similar to this on the previous exercise. And this has no statistical importance, it's just for graphing purposes. Okay, so if now you come here to the next chunk, this is where we're going to make a plot. I left you some active code. So the ggplot that you see there is active. If you run it, it's going to work. I left you some codes that I commented off because they're not the main part of this. But right now you can leave them commented like they are. I will tell you when to uncomment them. So here we're basically setting our rows and columns like we already did. Let me just decrease my font size so you can see. But this is just defining the imaginary physical space that this study will be implemented. So now if we recall from the previous times that we made this plot, we used a geom tile to create those plots. So let's begin by, add a plus, let's begin with geom tile. Oops, tile. And the first one that I want to bring is going to be the potassium rates. So we already specified the data on the top on the ggplot call. So we're just going to call the AES function now. I want to say color equals to k rates. kg per hectare. So the color of the cells will represent the potassium rates. I do want to change a couple things here before we run this. I mean, if you were to run it, it will look like this. I do want to make a lot of changes here. So if it's still within geom_tile, but outside of AES, add a comma. I want to first say show.legend equals to false. So the legend does not appear. Add a comma. I want to make the size. I think it's actually, we can try size, but maybe it's line width. But let's try size for now. So the width of the line around the tile is a little bit larger. I want to change the line. So there's an argument called line type. I want to change the line type to number 2, which is a dashed line, to kind of more closely match what we had in the slides. And finally, I want to say fill equals to NA. So it does not have a fill, just a color of the outline, so we can see through it. If you run that, you should be seeing this, where it's not exactly the same as we had in the lecture. As far as like, so in the lecture, the first 3 plots had just one outline around all of them. Here in ggplot, it's just putting the outline around each one of these smaller plots, but you can still notice that the first 3 plots are one potassium rate, the next 3 are another one, the next 3 are another one. So it's still randomized the same way. It's just the way that's showing here is not 100% like we did in the lecture, but still the same concept. Okay, so the next step now is I want to bring in our nitrogen rates and show them in this plot. So we're going to come here, add a plus at the end of the geom_tile. I want to call another geom_tile where we open the AES function, and I want to say on this one, fill equals to n rate kgHA. So if we just run that, you would see this. I do want to make a few changes here for us to implement. So the first one is so right now, if we look at the data set is coming in, both nitrogen and potassium rates are being seen as double. For the potassium rate, that's not going to matter. But for the nitrogen rate, to make it look more like what we had in the lecture, I want to treat nitrogen rate as factored. I can do that just on the go here. So not save that change to the data set, but just doing on the go within ggplot. If you come right before nitrogen rate, we can just use this function called factor. And make sure you add another parenthesis at the end of this line, so you close all parentheses there. So if you treat nitrogen as a factor, now it's going to give us colors like this, like discrete values of colors. So if you go outside AES, but still inside geom_tile, add a comma, hit return a few times. I want to change 2 things here. So one is I want the color of this tile to be black. And in this case, color relates to the outline color. I want to give it a comma. And I also want to say show legend equals to false. So this does not appear on the legend. If we run that, this is what we get. And I do want to remind you something here about ggplot. So ggplot works sequentially in a way that it first plots what you ask and then keeps adding the layers that you're asking from that point on. What we did here is plotting potassium first and then nitrogen. We're kind of hiding away the potassium lines, right? That we were able to see.
Scroll back up, Dr. Bastos.
Yeah, say that again.
Can you scroll back up real quick? I'm sorry. I just wanted to catch up real quick.
So the only thing, so if I want to see again, be able to see again the potassium rates, we just need to change the order. So we need to first bring nitrogen and then potassium. I did not want to show you that to begin with, because that's not the sequence that makes sense when we think about the design. So I want you to think about the design sequence. And then for plotting purposes, we're going to change them just to be able to see. but still creating that intuition of why we're doing this. So if you just copy the geom tile that we just did for nitrogen rate, you can cut it. And you can bring it right after that ggplot call and paste it. Remember to add a plus after it. And right now we have a plus after the geom tile for the potassium rate. You know what, let me do, so I want to take a quick moment here, stop here and run this, just so we see the effects of that. This is how it looks like. We probably have 3 more minutes in class. We are over time because we only go to, or we go to 11. Okay, so we're not, I'm sorry, I teach 2 classes and I'm always confused between the times. So we still have time, no worries then. So if you did that, I left you the remaining of the code here of what I want to do. So add a plus at the end of that geom tile for potassium.
One step back. So we have to.
Yeah. So the way it was before, let me just undo here. So we have first added the geom tile for potassium rate and then for nitrogen rate, which makes sense for us statistically. However, graphically, we are hiding the potassium rate outlines. So to fix that, we just need to bring potassium rate after nitrogen for plotting purposes, right? So I just copied the nitrogen geom tile or cut it, and then I paste it above the geom tile for potassium rate and added a plus so it connects. And for running this to show, I just removed that plus at the end of the geom tile potassium rate, because if you don't, then it's not gonna work because it is expecting something afterwards, and if you're not providing it, it's not gonna work. So it looks like this. So just gonna leave this here for one quick second. And then the next step here would be for us, I want to change this the color of the nitrogen background. So instead of blue, green, and red, I want to have the colors that I showed you on the lecture, which were 3 shades of green. So we can just add a plus here and then uncomment the beginning of the scale fill and the three lines with the scale fill. Notice that it has a plus at the end. So if you want to run just that, you have to remove the plus and run it to see the effect of that. So this is how it will look like. Now make sure that you add that plus back again, because we're going to uncomment more code. So the next one is just adding a geom text for the split plot label. So we have the split plot label. I just want to do that. I'll remove the plus so we can see just the effect of that. So we're adding the split plot label on top. Now I'll come back to the code, re-add the plus, uncomment the next one, which I'm adding the label of the treatment name. If you want to just run that, remove the plus so it doesn't affect it and run it. So this is what we see. Finally, I want to give the plus back again and uncomment the labs, which I'm just changing the title, and then uncomment the theme void, which makes the theme more clean so we can see the plot layout. So this is what you should see in the end. Perhaps it looks a little bit different than mine just because, again, the randomization was different. But to make sure that we all have the same output here, you would go back to the code chunk where we did the first randomization, which was right here. So the WP RCBD rand, you would run that. And then immediately after that, you run the next chunk, and then you do the join again. You don't have to inspect block 1, that's not assigning anything. You run again that plot layout chunk, and then the plot. So if you were able to do that, you should have been able to reproduce the same plot as I did here if you set seed the same way as I did. So, guess wrapping up here again, we first randomize whole plots to block first, and then we randomize nitrogen rates to whole plot experimental units, which were associated with the different potassium rates. So we can kind of see here that the three plots are one potassium rate, and within them we have three different randomized nitrogen rates. And then the next three plots are one potassium rate, again with three different nitrogen rates randomized, and so on. And that happened for all the blocks, right? So the difference in the experimental size and unit, and also the fact that we're randomizing with these restrictions in place, is what sets this up as a split plot design that we need to account for in the analysis properly. I have some notes here, just talking about that each potassium rate took 3 cells and each nitrogen rate took 1 cell. So again, that difference of experimental material being required. I think I left you here all the code just to export this as a CSV. So exporting the SPP RCBD rand, which would be your field, like your field layout if you go out and collect samples, right? Because now you have your rates, you have your blocks, your whole plot number, your split plot number, your treatment names, and then you could just simply add columns to this data frame to be the response variables that you measure on these plots, right? So plant height, yield, whatever you're measuring, you can just use this as a template. So I left you the code here to export this, and then just exporting that GG plot to file. So I left you the code already because we have already done something similar. So if you come to the Files tab, to the R button, and you go on your output, you should see your plot layout, which is the one that we just created, and also the CSV, which if you open here in R is going to try to bring in as a column like this, which is not very useful. But if you do open this through your actual file management system, it would open as an Excel file on your side. Okay, so we actually did not have time to do the exercise, so we're going to do that next class period. So you already have the script. We will also, I believe, next class is where we have the repeated measure class. We're going to start off with a lecture to define what it is, when it's used, and how it's done. And then we're going to get into an exercise of running repeated measures as well. I think we will be able to get to the actual exercise by next class, but for sure, we're going to finish this split plot analysis first, go through the repeated measure lecture, and then start the exercise. Okay, so before I open for questions, I just want to go ahead on GitHub and open for attendance. And if anyone has any questions while I'm doing this, feel free to ask them. Okay, so I just opened an issue, claim your attendance if you were in class. And also a quick note here, I did update the website links a lot last night. I know that up until then, I had not updated the link for the reading on the blocks random or fixed. I did fix that now, so you should be able to see that paper. That paper, it is just a paper that you can, it's a PDF, so you can download it if you wish. And I think even has my highlights now that I look at it. But anyways, you have access to that. I also posted another reading here, which I will send you more information. Let me just close my email. I will send you more information to read this, but it is a repeated measure reading. If you open this is actually a book, a PDF version of a book that's called Mixed Effects Models in S and S Plus. I think this has like, I don't know, 600 pages or more. So it's a very large book. I do not expect you to read everything. This is a good resource if you want to learn more. I think it's going to go into more detail in the math side than we're covering this class. So if you're comfortable with that, feel free to use this resource. If that is too complex, do not worry, because one of my goals here is to simplify some of those things and be more an applied way. But I will ask you to read the chapter related to repeated measures, so you create some understanding of that before the class, but I'll send you more information about that by email. And also, just for your reference, Pinheiro and Bates are, so they wrote this book, but they are also the authors of LME4, NLME, and maybe other packages I don't remember now, but they're really big names when it comes to running statistical analysis of experimental design studies in R. So these two names, Pinheiro and Bates, you're seeing a lot. They're the authors of this book. Really good book. It has a lot of examples with R code that you're going to be able to relate. And even if something that we cover in class that we do not go in a lot of depth that you would like to learn more, this is the resource I use always. So it's a really, really great resource for you to have handy. I would highly recommend you download this PDF here and keep close to you as you go through grad school, because it could help you in many ways. All right. I think that's all I had. Any questions? All right, I don't hear anything, so I'll see you on Thursday. Thanks, everyone.